Yeah, they're not, the Chesapeake Lake, because people go boat fishing for gar there. I have a boat fishing thing, but I've never used it before. I have a reel and a compound boat.
So everybody, we were talking about the blood, right? And in last lecture, I stopped by talking the process that is called hemostasis. Hemostasis is the arrest of bleeding or stoppage of bleeding. Remember that? Hemostasis, if you get a cut, then within few minutes usually bleeding stops, right? So a clot is formed there. So that process of stoppage of bleeding or holding of bleeding is called, or haltage of bleeding is called hemostasis. Now, hemostasis occurs in 3 steps. Number 1, I mentioned, is what? Vascular spasm. That's the constriction of the lumen of the artery. If you get a cut, this is the artery. If you get a cut here, the size of the lumen gets narrowed by constriction of the lumen. This happens due to pain reflex at the site of injury or cut. So that pain reflex causes the contraction of the smooth muscle in the wall of the artery, and that causes the constriction, that is the vascular spasm. So bleeding reduces. Number 2, you know the platelets love the rough surface. So platelets start to get attached and release the chemicals that make the surface of the platelets sticky. So more and more platelet will get together, and that is called what? Platelet plug. Formation of platelet plug. Okay? So they will get together, platelets, and will make a plug to seal the opening. And then number 3 is coagulation. In coagulation, what happens? You remember one type of plasma protein that is called fibrinogen or fibrinogen, one type of plasma protein. That plasma protein is converted to fibrin threads, thread-like structures, and they get together to form a mesh that is called fibrin mesh. Okay, like network of fibrin threads. Okay, they form a network. So these are fibrin threads. So fibrinogen, this plasma protein, forms the threads. Okay, and the threads go in different directions, okay, and get attached to each other and form the mesh. And this mesh now will do what? Will cover the platelet plug like this to make it stronger. So that is the clot formation. These 3 processes take place during the clot formation or hemostasis. Okay. So you see, first the lumen gets narrow, that's the vascular spasm, and then platelets get together to form a plug, and then fibrin mesh is formed, to cover the clot. Just know that platelet plug formation occurs by positive feedback mechanism. What is that? First few platelets get attached to the rough surface, right? Then more and more get attached and size becomes what? Bigger. So that is called positive feedback. Starting, from small, gradually getting bigger. Okay, more and more platelets are getting together. So positive feedback mechanism. And in last class, I mentioned that two chemicals are released from the platelets that make the surface sticky. One is ADP, another is thromboxane A2. I wrote on the board, you have it in your note. Okay. Now, how the fibrinogen, this plasma protein, is converted to fibrin threads. That is the coagulation, number 3 here, coagulation. So we'll talk about the coagulation more detailed, okay? So this number 3 is coagulation. Fibrinogen is converted to fibrin. So we'll talk about this process more detailed, okay? First, coagulation can occur in two pathways. How many? Two. One is called extrinsic, another is called intrinsic. So these are two pathways, extrinsic pathway and intrinsic pathway. For extrinsic pathway to start, you need tissue factors. Tissue factors. What is that? Let me tell you. If you get a cut, okay, your tissue gets injured, right? Like your muscle, skin tissue get injured. And the tissue will release some chemicals at the site of cut. And those are the tissue factors that will trigger the which pathway? Extrinsic pathway. So it needs tissue injury and release of those factors. Okay? Intrinsic. To activate, you need platelets factors. That means platelets should release certain chemicals to trigger the intrinsic pathway. Now you tell me, sometime you know that blood clot occurs inside the artery, right? Without any external injury. Blood clot can occur inside the blood vessel, right? So that will be intrinsic or extrinsic? Intrinsic, because no tissue injury, external tissue injury, right? That is intrinsic pathway. Okay? Platelets release certain chemicals and that will form the blood. Now if you get a cut from due to outside injury, that will release the tissue chemicals, right? Tissue factors. So that is extrinsic pathway. When we get a cut, right? That releases, tissue releases chemicals. So that is extrinsic. Make sense? So those are two pathways. Now there are total 13 coagulation factors or clot factors present in your blood. How many? 13. Many. 13. Coagulation or clot factors are present in the blood. And those clot factors or coagulation factors are important for blood clot formation. Make sense? So if someone has like lack of one or two or more coagulation factors, then what will happen? I said those coagulation factors are important for blood clot formation, right? Or coagulation. So if someone has like genetical disorder, someone has lack of certain factors, has like 10 or 11, 2 are missing or 3 are missing, then blood clot formation will occur, but it will be delayed. Make sense? Slow. The process will be slower. Okay? So that happens, bleeding disorders. Many types of bleeding disorders occur due to lack of one or more of those 13 coagulation factors. Okay. Among those 13 coagulation factors, first 4, 1, 2, 3, 4, we indicate the coagulation factors by Roman letters, numerals, like this. Coagulation factor 1, 2, 3, 4, like this. Okay. Up to 13. So first 4, 1, 2, 3, 4 are called the essential factors. That means what? Those 4 must be present to form the clot. Is it clear? Must be present to form the clot. Without any of those 4, blood coagulation or clot will not occur. Others, as I said, if 1 or 2 are missing, then it will be delayed. Make sense? But these 4, first 4 are what? Essential factors. Okay? See, so if any of these are missing, then blood clot will not occur. Just know that. So how many factors total we have? 13. Okay? They have different names. We will not go into detail. Just what is important here is extrinsic pathway will go through a number of reactions. Cascade, we say cascade of reaction. One reaction will stimulate the next one. Okay? So one reaction, the product will stimulate the next one. The next one will stimulate the next one. Okay? So cascade of reactions and different reactions in extrinsic and intrinsic. But finally, both pathways of reactions will produce a common product that is called prothrombin activator. Okay? This is the product of both extrinsic and intrinsic pathways. Although the reactions are different in extrinsic and intrinsic, okay? But the final product will be the prothrombin activator. So tell me, prothrombin activator will activate what? The prothrombin. That's why it is called what? Prothrombin activator. Make sense? That means it does what? Activates prothrombin. That means prothrombin is inactive, right? It should be activated. So prothrombin activator, now you know, does what? Activates inactive prothrombin, which is inactive prothrombin to active thrombin. So this is inactive, this is what? Active. Make sense? So, to convert inactive prothrombin to active thrombin, you need what? Prothrombin activator. Is it clear? Okay. So now this is active, right? This is active. So thrombin will do what? You see now. Will convert the fibrinogen to fibrin, which is thread. Okay. So you see, prothrombin will be converted to active thrombin by prothrombin activator, and thrombin will convert the plasma protein fibrinogen to fibrin thread. Okay? Then what will happen? I explained, right? The threads will get attached to each other, right? To form the mesh. Is it clear? And the mesh will cover the clot. Okay? So that's the process of coagulation. So coagulation takes place in how many pathways? 2. One is extrinsic, another is intrinsic. Okay? Now let me ask you, if without any external injury, blood clot is formed inside the artery, that is which pathway? Intrinsic. Make sense? if you get an injury, tissue injury occurs, right? Tissue factors are released, that will activate the extrinsic pathway, okay? Now, if I take the blood from your artery without injuring any tissue, okay, and put in a glass tube, blood clot formation will occur, right? If there is no tissue injury, no tissue factor is there. Make sense? So that clot formation will be by which pathway? Intrinsic. Make sense? Although outside of the body, but that is the intrinsic pathway. Okay. How many clot factors we have in human body, blood? Altogether? 13. First four are called what? First four are called what? Essential, everybody. Okay? And lack of clotting factor, if any of those first four is absent, then no clot. Make sense? Those are essential. If one or more of other factors, other 9 factors occur, then the clot formation will be slower, delayed. Make sense? But clot will be formed. Okay? Bleeding will be prolonged. Make sense? So that's the bleeding disorder. Okay. And what is the common product of both extrinsic and intrinsic? Prothrombin activator. That activates the inactive prothrombin to active thrombin, right? And thrombin then converts the plasma protein fibrinogen or fibrinogen to fibrin threads. Make sense? Okay, so that is the process. So here you see the intrinsic pathway goes through different reactions, cascade of reactions. Extrinsic pathway, the final product of both pathways is, or common product is prothrombin activator. Then prothrombin activator converts the inactive prothrombin to active thrombin. Thrombin converts the plasma protein fibrinogen to fibrin. Okay. So here you see the fibrin mesh. These are fibrin threads. Okay. Some red blood cells are trapped in it. After clot is formed, clot is a semi-solid structure. Semi-solid structure. What happens? It's like soft jelly-like structure, the clot, okay? Then what will happen? Retraction. The clot will squeeze, get smaller, and fluid will come out from the clot. So, clot retraction. So this is the clot, okay? It will get smaller, okay? And fluid will come out because it is semi-solid structure. Fluid will come out. That fluid is called serum. And the serum is like a plasma protein, but since clot has been formed here, the clot factors have been used up to form the clot. So serum is actually plasma protein, sorry, not plasma protein, plasma, the liquid part of blood, minus clot factors. Because clot factors have been used up to form the clot. Okay? So plasma, the liquid, but clot factors are absent there in the serum. So that is the serum. You remember in ANP1 you have learned the muscle contraction. Do you remember actin myosin? Okay. So inside the muscle fibers we have actin myosin, right? They cause the contraction of the muscle. Similarly, inside the platelets we have actin myosin, the contractile proteins. So after the clot is formed, platelet aggregation, right? Platelet plug, what happens? The platelets contract. So the clot will get smaller, make sense? And the liquid will come out, that is the serum. The liquid that comes out from the clot. disorders. Bleeding disorders could be thromboembolic disorders and bleeding disorders. So those are two types of disorders of hemostasis. Thromboembolic disorders. Thromboembolic has two parts, thrombus and embolus. That's why together called thromboembolic. So thrombus and embolus. What is thrombus? If blood clot is formed inside the artery or a blood vessel and stay attached to the wall. So this is fixed clot. Fixed clot attached to the wall of the blood vessel. Okay? That is called thrombus. Sometimes what can happen, you see, because blood is flowing, right, inside the artery. Blood is flowing. So due to that pressure of blood, this clot may get detached from the wall and start to move with the blood. Make sense? So this is free-floating, freely moving clot, right? Because it is no more attached to the wall. And that is called embolus. So freely moving clot. Clot is called embolus. If it is fixed attached to the wall, that is called what? Thrombus. If it get detached and move, circulate with your blood, that is called embolus. Make sense? Sometimes, if any air bubble is formed or go from outside into the blood, that also moves. The bubble, that is also known as embolus. So any moving, thing that can block the blood vessel could be termed as embolus. Now, embolus could be very dangerous because, if any clot is formed in your lung, or sometimes it happens in leg veins, the veins of your leg, most of the cases either in the lung or in the veins of the leg, they are formed, okay? And then they start to move, right, with the blood and can block the artery of your brain or the heart. Can go there and block, right? So that can kill the person, that can cause stroke or, coronary artery, blockage of the coronary artery. It's heart attack, myocardial infarction can occur. Okay, so make sense? So that is the thromboembolic disorder, thrombus or embolus formation. Bleeding disorder, that usually occurs due to lack of the clotting factors that I have mentioned. Prolonged bleeding, okay? Clot formation is delayed, right, or slow. So that can cause prolonged bleeding. So that is related to clot formation, delayed clot formation. Now, how you can prevent clot formation? There are some chemicals, aspirin, that inhibits thromboxane A2. How many of you have heard this name, thromboxane A2? Remember? Did you hear this? I mentioned in last class as well as today. Okay, so two things make the surface of the platelet what? Sticky. Remember that? ADP and thromboxane A2. So this is right. So we know that if the surface of the platelets get sticky, then they get attached to each other. So aspirin inhibits thromboxane A2. Is it clear? So the surface will not get sticky. So prevents platelet plaque formation. Okay, that's why you will see sometimes doctors, prescribe, take mini aspirin, especially older people, right? They will ask to take mini aspirin. Have you heard that regularly? To prevent clot formation inside the blood. Okay. Heparin is a natural anticoagulant. You must remember, I mentioned in last class when I was talking about 5 different types of white blood cells, basophil releases heparin. Make sense? So basophil is the main source of heparin. Another type of cells also release heparin, that is called mast cells in the tissue. So anyway, so basophil, one type of white blood cell, and mast cells, they release heparin, which is an anticoagulant, natural anticoagulant, because formed by the living cells, and prevent the clot formation. Heparin is also used in the lab to keep the blood liquid. You remember when you did the hematocrit? You did it, right? Those narrow capillary tubes, red markings, that means heparinized. They have heparin inside. Okay, so when the blood will get in, it will stay liquid. You can centrifuge to separate. So heparin is a natural anticoagulant. Warfarin is an artificial anticoagulant. Warfarin. Blood groups or types. Human blood has different groups or types. One is called ABO system, that includes blood type or group A, group B, group AB, and what? O. These belong to ABO system. Type A, type B, type AB, type O. Okay? And RH system includes positive and what? Negative. We know positive blood, negative blood. So that is RH factor or system. So we need both to indicate the blood type, right? We say A positive, A negative, B positive, B negative, right? So we need both. Now, why the blood type is different in different people? Blood type is determined by the presence or absence of antigens on the red blood cell surface. So this is a red blood cell. Very simple. What type of antigens are present on the red blood cell surface? So this is a red blood cell, okay? And if on the surface of the red blood cell you have A antigens, your blood type will be A. So what type of antigen is present, right? A is present only, so blood type is what? A. If someone has only B, no A, okay? B antigens. What will be the blood type? B. Make sense? If someone has both A and B antigens, blood type will be AB. If none of those, then blood type will be O. Is it clear? So if only A is present, blood type is A. If only B is present, blood type is B. If both A and B are present, blood type is AB. If none of those, O. Make sense? So, determined by the presence or absence of antigens on the red blood cell surface. Is it clear? Which ones are present, which ones are absent? Now positive and negative, that are determined by another type of antigens, and those antigens are called D. D antigens. If someone has D, that is positive. If no D, that is what? Negative. Is it clear? So now you tell me, this one has A, B and D. So what is the blood type of this person? AB positive. Make sense? If no D, AB negative. Make sense? You get it? So D determines positive or negative. Okay? Which is also called RH. You see here, RH. This antigen was first found in rhesus monkeys' blood. That's why the name came from rhesus monkeys. RH. Anyway, so very simple. That's how the blood type is determined. Okay. Now, what is important is this is your blood, red blood cells. On the surface, you have your blood type is A. So what type of antigen should be on the surface? A. Makes sense? Your blood, this blood type is A type blood. So type or group A. So these are A antigens on the surface of the red blood cell. Is it clear? Makes sense, right? Okay. Now, in the plasma, the liquid part of blood, you have antibodies in the plasma. Let's go back. On the red blood cell surface, you have what? Antigens, right? And in the plasma, you have antibodies, okay? Now there is a rule. What is that rule? If any antigen is present on the red blood cell surface, same type of antibody must be absent in the plasma. I am repeating again. If any antigen is present on the red blood cell surface, same type of antibodies must be what? Absent in the plasma. Is it clear? If same type of antibodies, look at this. This is blood type A, that means A antigens on the surface, right? If same type of antibodies are present, A antibodies in the plasma, antigen-antibody reaction will occur. Make sense? Same type of antigen-antibody reaction will occur between these two. And that will destroy the red blood cells. Make sense? That is called agglutination. Okay? That will cause the destruction of lysis of the red blood cell. So same type of antibodies must be what? Present or absent? Absent, because it is dangerous. If same type of antibodies are present, that will cause agglutination. So this is blood type A. On the red blood cell surface, you have A antigens, right? All blood cells, red blood cells, you have A antigens. In the plasma, you should not have A antibodies. Is it clear? So what type of antibodies can be present? The opposite one, B. That is the rule. The other type of antibodies is safe. No reaction will occur. Okay. So now you tell me, this is blood type what? A. Is it clear? Because you have A antigens on the red blood cell surface. Let me draw another blood type. This is another person. This person on the red blood cell, you have B antigens. So what is the blood type? This type? B. Make sense? Okay, so this is type B. In the plasma, what type of antibody should be present? A. Make sense? The other type. So in the plasma, you have A antibodies. That is safe. If same type, then that will cause the reaction. Okay. So this is type B blood. This is type A blood. Okay. Now, if you give this blood to this person is the recipient, and this person is what? Donor. Okay. So donor's blood enters into the recipient's body. Okay. Now you see what type of antibodies in the plasma. Okay, right? And what type of antigens on the red blood cell of this blood? A, right? So when this A blood will enter into this B type person's body, he has huge amount of A antibodies throughout the body, right? In the plasma, this person. So these A antibodies will quickly destroy these red blood cells, because same type of antigens on the red blood cell surface. Agglutination will occur. Make sense? Because when these red blood cells will enter into this body, in the plasma of this blood has huge amount of what? A antibodies. Make sense? Will bind with these antigens, will destroy the cell membrane. Okay, that is the mismatched blood transfusion and reaction. Yes.
Okay, so with O, if there's no antibodies on the O type blood, then
No, AB type blood.
Well, I'm sorry.
In O type blood, now you are talking about O, right?
Oh,
so
right.
let's draw first, then it will be clear. So you are talking about O type, right?
Right.
So no antigen.
Antigens.
That means both A and B antibodies are present, right?
Okay.
Yes. So no antigen, no antigens on the red blood cell surface. That's why it is O, right?
Okay.
No A, no B.
Right.
Now you tell me, if this blood enters into this person, since on the red blood cell surface no antigen, whatever antibodies are present will not destroy the cells.
So that's why O is a universal.
That's why O is universal donor. Make sense? Because on the surface of red blood cell there is no antigen. So antibodies will not be able to destroy the cell membrane. So you can give O to this, you can go O to this, these antibodies will do nothing, these antibodies will do nothing. Is it clear? Okay. So that's why O can go to any blood. Now if you give B blood to A type, same problem. On the surface of red blood cell you have B type antigen, in the plasma you have B antibody. These antibodies will destroy these red blood cells. Make sense? Okay. Now let's talk about the other type, AB. This is AB blood. So you have both A and B. Okay, so you have A antigen, B antigen. Okay. Now AB is called universal what? Recipient. O is universal donor, AB universal recipient. Okay. So now in the plasma, do you have A antibody? No. Do you have B antibody? No. So no A or B antibodies in the plasma. Make sense? So if you give A type blood, we know that on the red blood cell surface we have A antigens. If you give B type blood, we have B antigens. Make sense? Can easily get into this blood because no antibodies here, right? no antibodies here. So we will not be able to destroy these cells. Okay. Now, remember one thing, another rule. Another rule is very important, that when we do blood transfusion, always we consider the red blood cells of the donor and the plasma of the recipient. We consider the red blood cells of the donor and plasma of the recipient. Why? Because you see, this one, AB, has no antibodies, so we will consider the red blood cells of the donor, this person's, and the plasma or antibodies of this person. So this is safe. Now, the problem is if you do this way. Let's go back again. This is A type blood, A antigens. What type of antibody you have? B. Make sense? Antibody in the plasma. And this is another person. This person has B type blood, B antigens, and in the plasma you have what type of antibody? A antibody. Now, if I give this one to this blood, the problem is antigen of red blood cells, right? And antibodies of this, right? But if you think this way, this person has what type of antibody? B, right? Because A type blood, B antibody. Is it clear? Why we are not thinking that this B antibody can destroy these red blood cells, the recipient's red blood cells? In this case, these antibodies, the donor's antibodies, will not be able to destroy the recipient's red blood cells. Only when the donor's red blood cells will enter, they will be destroyed. Donors, the blood that is going into recipient's body, those red blood cells will be destroyed. The donor's red blood cells, not the recipient's red blood cells. Why? Because you see, the amount of the plasma of the recipient, like this is the antibodies. If you give this blood here, these donor's red blood cells will be destroyed, but this antibody will not be able to destroy the recipient's red blood cells, although same time. Although the same time. The reason is the antibodies of the donor will get diluted in a huge amount of blood of the recipient's body. The antibodies will get what? Diluted. So it will get very weak. So they will not be able to harm the red blood cells of the recipients. Okay? So that's why we always consider donor's red blood cells and recipient's antibodies or plasma. Okay? That is the rule. Okay. Erythroblastosis fetalis. Erythroblastosis fetalis is a clinical condition of fetus or newborn baby. Now, the name tells you erythroblastosis fetalis. Erythroblasts, what type of cells? Yes, erythro, red blood cells, right? Blasts are immature. That means in newborn baby, in the blood, you will find many immature red blood cells. That's why this clinical condition is called erythroblastosis fetalis. In fetal blood or newborn baby blood, you have plenty of immature red blood cells. Which is good or bad? Immature red blood cells have nucleus but no hemoglobin. You remember? Hemoglobin appears during maturation. So immature red blood cells have nucleus but no hemoglobin. So if red blood cells, you have immature red blood cells in your circulation, they don't have hemoglobin, are they useful? No, because we know that hemoglobin is that, right, component that transports oxygen, right? That's the function of red blood cells. So immature red blood cells are not able to or capable to transport oxygen. Now, if in fetus or newborn baby's blood has plenty of erythroblasts, immature red blood cells, that blood will not be able to transport oxygen enough oxygen. Make sense? So that is the problem, erythroblastosis fetalis. Now, why the newborn gets erythroblastosis fetalis? If the child or fetus is Rh